Good morning. It's a pleasure to be here to share with you some recent observations that we have done in the last year. And I'm going to talk about new chemoreceptors in the central nervous system. But first, I would like to have a look on what we know about the olfactory system. And the olfactory system is represented in the olfactory bulb as the primary organ, in which several neurons are the sensory cells, which are specialized in the recognition of different ligands, odorant ligands. And these receptors are found, these neurons have specific receptors. About 1,000 of these receptors are present in rodents, and about 400 receptors are present or are putative receptors in human. And all the system is a very complex and very sophisticated one, because receptors bind to specific ligands and then produce the signal and transmit the signal through adenyl cyclase, specific adenyl cyclase, which is called adenyl cyclase 3. And in addition, it is involved also a kind of G protein, which is called G-alpha OLF. And in addition, we have certain transporters and shuttle proteins which permit the movement of these receptors from the cytoplasm to the cell membranes. Then the signals arrive to specific cells at the glomeruli, and then to mitral cells, and then all this system sends the perceptions from outside to the primary and the secondary centers in the nervous system. And in the context of this meeting, we have to recall that several neurological diseases, for example, Alzheimer's disease, Parkinson's disease, schizophrenia, and many other neurological and psychiatric diseases, one of the main aspects of this disease is the involvement of the smell. So patients first present at early stages of the disease disorders in olfaction. Well, in this context, and completely different of this first point, we started the work by analyzing, by assessing the expression of different RNAs in the cerebral cortex in Parkinson's disease. And this was within a project which is trying to produce some kind of data in relation with the combination of different omics, transcriptomics, proteomics, metabolomics, lipidomics, etc. And we have found that it was a difference between controls and patients with Parkinson's disease. This is in the cerebral cortex, and this is a heat map. And by processing this data with very robust bioinformatic analysis, we observe at this time that either using KEK or using gene ontology, two different pathways or molecules belonging to two different pathways were abnormally regulated in Parkinson's disease cerebral cortex. And these were the system olfactory transduction and the taste transduction. We were amazed by this data because we didn't know that it was a possibility that these genes were represented and RNAs were represented and expressed in the brain of patients with Parkinson's disease, and even less that these genes were abnormally regulated in the cerebral cortex of in these cases. Then we moved to the validation of this data and demonstrated that several olfactory receptors, at least the mRNA of these receptors, were abnormally expressed in the frontal cortex in patients with Parkinson's disease. And this was in contrast with the observation in the also in the cerebral cortex, in which taste receptors were also dysregulated, but in this case the dysregulation was mainly an upregulation, in contrast to the fact that the olfactory receptors were downregulated. Well, then we tried to understand what happened with these receptors and where these were expressed. And then we used immunohistochemistry just to show the expression of the receptors in different cells in the central nervous system, including the hippocampus, the internal cortex, the cerebral cortex, the cerebellum, and the brain stem, including also the substantia nigra. So these receptors are widely expressed in the human brain, and also the proteins which are necessary for the function of the receptors, that is adenyl cyclase 3 and the G-alpha OLF, which are crucial to the function of the receptors, are also expressed in the whole brain. In order to understand what was the subcellular distribution of these components, we employed RAFs, lipid RAFs fractions, and discovered that the receptors were mainly in the cytoplasmic part of the fraction. And this was also demonstrated by immunohistochemistry, showing that the main distribution of the receptors was in the cytoplasm. But what is important is that in addition to the receptors and the signaling pathway, we have also several shuttle proteins which permit, at least in the sensory, in the olfactory system, in the peripheral sensory system, the shuttle of the proteins which are in the cytoplasm to the cell membranes. And cortical cells, mainly neurons, have also the expression or express also the shuttle proteins which will permit the transport of proteins of the receptors and other proteins to the cell membranes, and also several other proteins which are involved and which are specific of the sensory, the peripheral sensory system, olfactory system, are also expressed in the brain. Then we move to the substantia nigra, and then in substantia nigra there was also the regulation of these olfactory receptors, and this was in contrast with the upregulation of taste receptors, which are also present in the brain in the human brain. So the deregulation is not merely the result of a decrease in the number of cells, but rather the expression of that something goes wrongly. And then we move to the other diseases in order to compare and to know what happened with in the cerebral cortex with other diseases. And these represent internal cortex in Alzheimer's disease at different stages of disease progression. That means at the stages 1, 2, 3, 4, and 5, 6 of Braak and Braak. And then we also observe the regulation of several of these olfactory receptors. And it should be noted here, for example, that there is not only a decrease in many instances, but also an upregulation of several olfactory receptors. And again, the receptors, which are mainly involved in the beta recognition, are also deregulated, and in this case, again, in the form of upregulation. What happens in the cerebral cortex? Something similar, but a bit different. So there are regional differences in the deregulation of these genes in the cerebral cortex. For example, here we see an upregulation of olfactory receptors in the brain. And then we tried to understand what happened with other species, and in different models, or at least in one of these models of Alzheimer's disease. And then we used the APP1 transgenic mice as a model of Alzheimer's disease, in which several plaques appear at 3 months of age, and behavior and cognitive impairment. We can talk about cognitive impairment in these animals occurs at the 6 months of age. And what happened here is that, again, the receptors are expressed in the brain in the cerebral cortex, in the brain of mice, and also the signals or the molecules which are necessary for the function of this system, at least in the peripheral sensory systems. And again, the olfactory receptors were altered. The expression of these olfactory receptors was altered. In this case, you can see here that there is not a decrease, but rather an increase in the expression of the receptors. Other diseases also have this kind of abnormalities. For example, in progressive supranuclear palsy, you can see here that the majority of the receptors are upregulated. That means that they are dysregulated in another sense. And the same happens with the T receptors. And also in Creutzfeldt-Jakob disease, that it is in this frontal cortex, in which we can see also differences between two subtypes, BB1, MM1, and BV2, which show differences not only regarding the disease, but also between two different subtypes, and also occurs in the cerebellum in Creutzfeldt-Jakob disease. So it's very clearly that there is a deregulation of these of the genes in the cerebral cortex in several human neurodegenerative diseases. And then we try to understand whether these changes were related with epigenetic factors. And then we studied by parasequencing the possibility that these genes were abnormally methylated in the different disorders. And we found that several genes were modulated probably with or by means of methylation of gene promoters, but this is not the only mechanisms, of regulation of olfactory receptors. The olfactory receptors are also present during development. Here is the brain of a fetus in which the receptors are expressed in different regions of the brain. So these receptors appear very early during development. Then we move to another approach, and this approach was mainly focused on the dopaminergic neurons in the substantia nigra. And this study was carried out in collaboration with many other people, and we used different methods. So in this case, in the previous case, we used just the demonstration of by means of RNA arrays. And now we move to a very potent tools as nano-cage analysis. And then we demonstrated the presence also of several receptors, olfactory receptors in dopaminergic neurons. Then we tried to understand the amount of knowledge about the amount of the receptors in dopaminergic neurons, isolated dopaminergic neurons in the rodent brain. And then by in situ hybridization, we really demonstrated again that these cells express these receptors. Then we also used the same method to know whether the receptors were only expressed in the brain or in other systems. And we were able to demonstrate that these receptors were present in several places in the central nervous system, but also in many other organs. Then we move again to the dopaminergic neurons, and then we try to demonstrate whether these cells were functional in the sense that the olfactory receptors expressed in dopaminergic cells or in transfected cells expressing these receptors were active, were functional in front of different stimuli. And by complex bioinformatic assays, we try to recognize different putative ligands to the receptors, and then we discovered that these putative molecules were putative ligands to these receptors. Then we selected some of them, a few of them, and then we exposed the cells to these odorants or these putative ligands. And then we demonstrated that these cells were responsible to these different stimuli. And again, that these stimuli produce changes in the calcium levels inside of cells. So clearly these cells were functional in the sense that they had all the mechanisms or all the molecules which are involved in the peripheral sensory system, and also they respond to different stimuli. So they are, in some sense, functional. So, in summary, what we have seen is that the olfactory receptors and obligated molecules which are needed to the function of the system are present in the central nervous system and in other systems in the human brain and in the mouse brain. And this has been demonstrated by nanocage, PCR, in situ hybridization, Western blotting, and immunohistochemistry. So we have a lot of information regarding the presence of these olfactory receptors in the brain. Well, these are also expressed in primary dopaminergic neurons, and these cells are also functional because mesencephalic dopaminergic neurons and transfected cells which express these the receptors respond to different specific stimuli. And an important point is that, starting again with coming to the beginning or moving to the beginning, that these cells are these receptors and differentially deregulated in several disorders. And these are not mere result or cell loss, but rather is a very specific aspect in these diseases. As I've said, the olfactory receptor regulation is only barely by DNA methylation of gene promoters. And in summary, or as conclusion, brain olfactory receptors are a new class or central chemoreceptors with a non-function and probably related with the contact or with the interneuronal signaling of different cells and throughout odorant-like molecules which are not really known at present in the central nervous system. And now I want to show you something else, because all these results are published or have been published this year or are in press, but now we have started to move to another model, which is the C. elegans, in which we have the possibility to study different the different related odorant or chemoreceptors which are related to these odorant receptors. And for example, we have seen that the cells which conform the oropharyngeal chemoreceptors, which is composed of 6 different or 6 neurons, are altered in mutants of Parkinson's disease. And most important, that the NMER, which is an homologue of these receptors, murin receptors, some of them, or at least one of them, the NMER4, produce the mutation of these, and NMER produce loss of neurons in this ring, this oculopharyngeal ring. This is a very amazing, a very interesting possibility, because these receptors appear at very early stages or at very primitive species, and are still present in the rodent and in the human brain. So we don't know at this point what could happen, what could discover in the future regarding the olfactory receptors, but this is just the first side to the presence of this new kind of receptors in the nervous system. And of course, you can imagine that this amount of work has been carried out in collaboration with this lot of different persons in different labs, including CIBERNED and including CIBERNED and including different hospitals here in Spain, but also in different places in all over the world. And particularly, we would like to stress the collaboration with Stefano Kostinczyk at the CISA in Trieste and Piero Caminci at the Riken Omics Science Center in Yokohama. And these have also been carried out with the help not only with CIBERNED, by CIBERNED, but also with these two European projects, which called Devil H and Dopaminet. And thank you very much for your attention.
Thanks, Isidro, for this beautiful talk. And I just have a question also about the striking olfactory receptors in cortical neurons. What about the normal function? And if you think that odorants are the exclusive ligands for these receptors. And also, there is also the vision that olfactory receptors play a fundamental role in axogenesis and axonal guidance and things like that.
Yes, you're right, the only experiment that we know at present about the possibility that these olfactory receptors act on different functions in addition to the olfaction is the axon guidance to the olfactory bulb. And we try to learn what happened during development, and we don't know at this point what happened during development. And the problem is that we don't have just one ligand. So the problem is that we have about 400 receptors in the brain. So it's very difficult to find exactly the ligand which produce some specific signal in the brain. Another possibility could be to knock out the G-olf in order to block exactly all of them. And there is one model which was produced some years ago, and these animals show a decreased olfaction, of course, but the authors say that these animals behave abnormally. So we don't know what is the meaning of behave abnormally, but these animals were not normal at all. So probably they have other functions in addition to olfaction. What is very clear is that they have a very crucial function, because there is a representation of about 400 genes in the brain.
Yeah.
And it's a lot of, imagine that vision, for example, needs just three genes. And olfaction needs about 1,400 of the genes are expressed in the human brain. So they are probably important, but the real function we don't know at present.
I think we have time for 2-3 short questions. Isabel.
Yeah, do you have any evidence that these receptors follow the rule of the allelic exclusion for the expression as in the olfactory mucosa? Whether once the neuron expresses one receptor, then the others are silenced.
Yes,
do you have any evidence that this happens also in the brain?
Well, this is well known in the peripheral sensory system, but it seems to me that several receptors are expressed by a single neuron in the brain. So one of the difficulties was obtaining good images by confocal microscopy, but we think that several receptors are expressed in the. So what it seems, and the theory or the hypothesis, is that receptors were at the beginning expressed in all neurons, and T receptors were also present in all neurons, and then with evolution, these receptors were moved to specific sites at the periphery to recognize different signals. So at the beginning was in the brain, and then the sensory organs specialize it from the brain.
Over there.
You see?
More questions?
This one there.
the puzzles in Parkinson's disease has been what shared feature do susceptible neuronal populations exhibit? And clearly it's not dopaminergic, since olfactory neurons are attacked early in disease. So I guess the question might be, does the expression of olfactory receptors identify the susceptible populations in Parkinson's disease? And I'm specifically thinking about the neurons that innervate the gut, which also fail very early.
Yes, this is a fascinating question, because we have not found already the link between the olfactory deficit in Parkinson's disease in relation with the present data. What is interesting, for example, is that several studies have been carried out in the epithelial, olfactory epithelia, and have found that terminals there have synuclein and also tau and amyloid in nerve terminals. But there is not a single study demonstrating the absence of the abnormal expression of olfactory receptors in the epithelia, and all the information that we have in relation with the loss of olfaction in Alzheimer's disease and Parkinson's disease starts in the bulb, because the majority of theories say that the accumulation of alpha-synuclein in the bulb and the involvement of the primary and the secondary centers in the brain are the cause of the loss of olfaction. And our hypothesis, at least as a working hypothesis, is that olfactory defects are the expression of a more complex deficiencies, which are expressed in the brain, in the cerebral cortex and in other parts of the brain, and also in the peripheral sensory organs. And the same happens with these receptors, because the main focus is on olfaction, but also it's necessary to have in mind that taste is also impaired in Alzheimer's disease and in Parkinson's disease. And what is important is bitter perception is altered in these disorders. And we have seen here that the receptors which are normally expressed are those related with bitter perception. So it is very amazing and very fascinating, but we don't know exactly at this point, at this moment, the real answer.